Tissue culture is the second method for the cultivation of animal viruses. So the first method was inoculation of the virus into an animal, right, in which we have seen different experimental animals, and we'll detect the growth by observing at the inoculated animal. Now the second method is tissue culture. This tissue culture or cell culture consists of cells grown in culture media in the laboratory. That is what we call a cell culture or a tissue culture. Just like how do we cultivate a bacteria, we can also cultivate cells or tissues. And these cultures could be propagated and handled similar to that of a bacterial culture. Right. So this will be more convenient to handle with these tissues and or cells than with an animal itself. So this like this is an in vitro living system, right? That means the living cells are there, but this is not in a living organism but outside a living organism. So there are different types of tissue culture systems available. They are given like organ culture, explant culture, cell culture. And within the cell culture, we have primary cell culture, diploid cell culture, and continuous cell cultures. This is the classification of tissue culture systems. The organ culture, as the name indicates, usually done for highly. Certain organ part of the organs are taken out, and the virus is inoculated in that tissue, and this is usually done for highly specialized parasites and viruses, right? You can given an example. Tracheal ring culture is done for isolation of coronavirus. Because this coronavirus usually they colonize in the tracheal system. That means in the respiratory system. So this tracheal ring culture is the tracheal tissues. Organ tissues are the main media or main source for the growth. So that is organ culture. This is not widely used, very rarely used. In case of specialized or viruses, these kind of systems will be used, and it could not be propagated further. That's only for one time usage. Then explant culture, it's like fragments of tissue can be grown as explant in plasma clot. That means tissue fragments are taken out. Not it may not be an organ, it could be a tissue fragment, and as an explant in plasma clot. This is also rarely used. Then third one is cell culture. It's the most widely used method, and we will be focusing on this one in detail. And the cell culture are of three types, as already stated: primary, diploid, and continuous cell lines. The primary cell culture of primary cell lines are these are the normal cells freshly taken from animal or human body, and these are cultivated, right? And usually the property is that they are able to grow only for limited time and cannot be maintained in serial culture. That is an important feature of primary cell culture. They are taken from a whole living system and they will be cultivated, and they are cultivated for only a limited period or limited cycle, not for a prolonged period or as a continuous system. And these are used for the primary isolation of virus and production of vaccines. Various examples are given like kidney cells, then kidney cell culture, human embryonic kidney, chick embryo cell culture. Oh, these are different examples. Then the diploid or the semi-continuous cell lines. These are diploid cells are taken out, and they could be subcultured to some extent. That means to around 50 times, approximately 50 times of serial transfer is possible, and after that they'll undergo aging, and that will be removed. Okay. And used for you especially used for certain fastidious virus, and of course production of vaccines. Examples given like human embryonic lung strain, then rhesus embryo cell strains. All these are different cell lines used for the specialized viral cultivations. Then the third one is the continuous cell line. It is actually, as the name indicates, it's a continuous cell line. You can maintain for longer years, and hence this is maintained. They have unlimited division capacity is there, right? And such a cell is what we know, it is a cancer cell, definitely. So these are derived from cancer cell. They can be serially cultured, and that's why it is called continuous cell line, right? So they are maintained as a serial, just like in bacterial system also, as serial subculture, or by storing at a deep freezer or at a very low temperatures. And they are one of the limitation is that since they are derived from cancer cells, they could not be successfully used for the vaccine production, because virus the cancer cells are not showing the genuine properties of a healthy cell. We have various examples like HeLa, human carcinoma of a cervix cell line taken from a lady. That's what the name indicates. Then Hep2, human epithelia of larynx cell line. Then BHK2, baby hamster kidney. All these are different examples for continuous cell lines. Again, in this slide also you can see different examples. Then HEL is there, Hep2 is there, RMK, different things are there. Okay. Then the advantages and disadvantages are like, for if you are using tissue culture, this is cheaper, sensitive, easy to execute, and have a broad spectrum of virus could be used. Then disadvantage is that we need an experienced technician, expertise is required, and also state of the art facilities are also required. A good laboratory facility is also required to avoid contamination, because tissue culture maintenance is the crucial thing in case of tissue culture cultivation or animal inoculation. Animal virus cultivation, if we are using tissue culture, the tissue culture maintenance is the key in that, right? So this is about tissue culture. So in the next segment we will see the third one, that means embryonated egg. Thank you.